Hi everyone, thanks for having me. I'm excited to talk to you today about some work that we've been doing in genome sequencing in kidney disease. Most of my talk will be about a condition that we're particularly interested in called autosomal dominant polycystic kidney disease. It's a bit of a mouthful, so I'm going to call it by its acronym, which is ADPKD. And I might start by describing this picture that we have here. And what we have here, this smaller image, is of a normal kidney, and you can see that it's got nice, shiny kind of outside with very smooth edges. And next to it is a kidney from a patient who has ADPKD, and you can see it's much larger than a normal kidney, and it's covered in these cysts. And these cysts develop through the lifetime of a patient and usually start to become a problem in early adulthood, usually in childbearing years. And they start to take over the normal architecture of the kidney such that the kidney doesn't work as it's supposed to, and these patients often need dialysis to survive or need a kidney transplant. And these patients usually develop renal failure relatively young, an average age of about 50. We hear a lot about dialysis, but it's a difficult treatment, and though it does its job, is not particularly good for you, and patients on dialysis have quite a shortened lifespan, often worse than a lot of cancers actually. This particular condition is really common. About 1 in 500 to 1 in 1,000 Australians are affected by this kidney disease, and it's the most common genetic cause of renal failure. About 10% of our dialysis population in Australia is on dialysis because of ADPKD. And even though it's so common and affects people's lives so dramatically, there's very little in the way of treatment for the condition, and there certainly is no cure. So polycystic kidney disease is a genetic disorder, and it's caused by mutations or mistakes in one of two genes that we call PKD1 or PKD2. And it's called autosomal dominant disease because it's inherited in families in what we call a dominant manner, such that it's passed through different generations. So a parent who has this condition has a 50% chance of passing on the condition to their children. Just a little bit of a recap of what you heard earlier from Marie about our DNA. And when we have a look at our DNA under the microscope, that it looks is packaged into little packets of information that we call chromosomes that line up nicely in pairs. And we have pairs of chromosomes because we get one from mum and one from dad. So we're 50-50 of our parents. And when we have a look at our chromosomes in more detail, they're broken down into little packages or pockets of information that we call genes. And our genes I like to think of as a very specific instruction for one particular function of the body. So maybe our chromosome is like a recipe book, and our genes are each of the pages in that recipe. And if we get particular mistakes in that recipe or in that gene, it can result in conditions like ADPKD. One of the other really stark things about this condition is that most patients with polycystic kidney disease don't actually know what the genetic basis of their disease is. And despite all of the advances that we speak about in genetic sequencing and you've heard about today, and despite how common it is, very few people know the underlying cause of their disease. You might ask, well, why is that important to understand the genetic cause of this disease? And I guess from a research point of view, I would say that if we don't understand the problem, we're never going to figure out how to fix it. And also from a clinical point of view, it's really important. When we see patients in clinic, these are some of the questions that we get asked. And this is a family of many generations with lots of people that are affected by ADPKD. We had a dad who passed on the condition to 3 out of 4 of his children, and then it was passed on to a number of his grandchildren as well. And we met one of these patients who wanted to know whether their children were going to develop this condition. This is another family with a mother who has a son who's also affected with the condition, but a number of younger children who were in early adulthood who still hadn't exhibited some of the features of the disease. And they wanted to donate a kidney to their mother, but we needed to make sure that they weren't also going to develop the disease. And to know that, we really need to know their genetic sequence and the exact mutation or mistake that's happening in this particular family. And one last family is a man with polycystic kidney disease who unfortunately had twin children who passed away as babies with polycystic kidney disease. And this is very unusual for this condition that usually develops in adulthood. And so they, of course, wanted to know what was happening in their family. And again, we hope that understanding the genetics in their own families will help us answer these questions. But sequencing in this particular disease is not straightforward at all. One of the problems is that the genes are really big, which can make it quite tricky to sequence. And the other thing that really is the major problem in sequencing these genes is that one of the polycystic kidney disease genes has something called pseudogenes. And pseudogenes are kind of something that's happened previously in evolution, where a little copy of that gene has been made 6 times in our DNA on the same chromosome. And that copy doesn't do anything that we know of. It doesn't produce any proteins, but it's very, very similar in sequence to the functional gene. And so sequencing machines have a lot of trouble telling the difference between the different sequences. Kind of like if you had a book with several pages that were very similar, and it's hard to differentiate between the two. And so at the moment, it's very costly and expensive to sequence these genes for that reason. But what our group has been looking at is using whole genome sequencing, which is a very new technique, and you saw some of the machinery in Dr. Thomas's talk, to see if that would be a better methodology to sequence patients with polycystic kidney disease. And we're kind of very uniquely placed in the world to test this particular experiment. We're in a facility that has very state-of-the-art sequencing equipment, and we're a group that's really interested in kidney disease. And really our overarching goal is to be able to develop a test in Australia that patients can access to know more about their disease for ADPKD. So as I'm sure you've seen some of these pictures before of the HiSeq X instrument that performs whole genome sequencing and is able to do it in lots of patients in a relatively short amount of time. And the thing about whole genome sequencing is, unlike other sequencing techniques that might sequence or decode very particular segments of the genome or particular genes, whole genome sequencing sequences everything. It sequences our entire genome and doesn't make any distinction between different parts of it. And so this is what some of the data that we look at looks like. It's really pretty colours, so I like it. But up here is one of our genes, and down here are all of the different reads from the sequencing machine that we stitch together to figure out what the unique genetic code is for this particular patient. And we started with about 40 families with polycystic kidney disease, and we performed whole genome sequencing on them to see if we could find a genetic diagnosis in them. And we were able to make a diagnosis in about 90% of the patients that we sequenced, which was really exciting for us because with previous technologies, people are able to make a diagnosis in about 60 to 80%. So with whole genome sequencing, we were able to find an answer for many more people and in a much more straightforward and accurate manner. And we were really excited to have some of this work recently published in the European Journal of Human Genetics. But what's much more exciting than having work published is what the answers of our research mean to the patients that are involved in it. And going back to the first family that I was talking about, we were able to find the particular genetic change which is here in this particular family, and it meant that we were able to test some of these younger patients in the family, the young adults in the family, and be able to tell them whether they were going to develop the disease or not, which as a young person is really powerful for planning your life and also doing as much as possible to try and prevent a disease from progressing. So I might take a little step back and talk a little bit about what we mean when we say that we find mutations or mistakes in genes. And as Marie mentioned, our genes are really, they're just a code, kind of like a computer code, of a series of letters in different sequence that our body is clever enough to decode and use to make particular proteins in our in all of our cells. And these proteins are used for all of the different functions in our cells. They might make up part of the walls of our cells, or they might make up some of the energy factor of our cells, but altogether the many different proteins make up the human body. And if we're to develop a particular mistake in our genetic code, then that protein isn't encoded in quite the right way. Maybe not enough of it is made, maybe too much of it is made, or maybe it's not made in the right shape such that it doesn't work properly. And that's what we look for in the genetic sequence to try and diagnose these patients. What is really tricky is, as Marie and Dr. Thomas were talking about, is that there's lots of variation that's just normal, that I don't look like you, because we all have different variations in our genome that are just part of what makes us us, but they don't cause disease. And a really tricky part of our research is being able to try and figure out what particular change or mistake causes the proteins to not work, and what change is just part of what makes us us. And so back to some of our other families, we were able to find the particular genetic change in this mum, and we were able to test some of her adult children. We found that this daughter here didn't carry that change. She hadn't inherited it from her mother, so she was able to donate a kidney to her mother, which is really exciting. And then our last family, we did find in this dad that he had a particular change in one of the polycystic kidney disease genes, but we're not actually sure whether that is enough to explain what was such severe illness in his children. And so there's more work for us to be done here to understand exactly what's going on in this family, and we hope that in all the genetic information that we have from having sequenced their whole genome, that we're going to be able to do that. And this particular condition, polycystic kidney disease, brings up lots of different unanswered questions. Actually, despite how common the condition is and how much research has been done, we actually don't understand why these particular mistakes in these particular genes cause the cysts to develop. And we hope that by sequencing more patients and being able to sequence the whole genome, that we'll be able to better understand that, because if we're able to understand what causes a cyst to develop, then maybe we could do something, could intervene to stop them from developing, or maybe have them develop slower so that there's more normal kidney tissue that's preserved. So there's lots more for us to do. One of our first main aims is to be able to get this research out to be able to be accessed by Australian patients with polycystic kidney disease. So we're working with the Kinghorn Institute to develop this test as an accredited test that will be available for patients to access. And by understanding the sequencing more, we hope that it will be able to allow us to investigate the reasons why cysts form, so that we can work towards cure and prevention. And I guess there's the other question about that 10% of patients that we didn't find an answer in. And we hope also that with the whole genome sequencing, which hasn't been done in this group of patients ever before in the world, that we'll be able to find an answer that hasn't been looked at before, perhaps another gene that wasn't looked at, or variation in some of those non-coding parts of the genome that we don't know a lot about yet. So there's much more for us to do, which is really exciting as well. I think that's all I had. I'm very lucky to be working with a really lovely team that do a lot of this work, and obviously all the patients that make it happen. But thank you very much.